Okay, our next speaker is Jayalekshmy from College of Agriculture, Kerala Agriculture University. She's going to talk about effect of microgravity and space environment on seed astroculture.
So, a very good afternoon to all. So, something very different I'm going to speak because this is only a proposal. We have not cleared for the Gaganyaan. So, it is the effect of microgravity and space environment on seed astroculture. So, as we have been hearing from the morning that the space environment provides a lot of different situations and environment situations like strong cosmic radiations, microgravity, weak geomagnetic field, and supervacuum. All these aspects, as we have seen, as it affects the human beings, it will also affect the plants. So, when we come to this, the two major aspects that affects the plant system in the space will be the effect of radiation and the effect of microgravity. So, as the duration of the space flight increases, the exposure time to the radiation and microgravity will also increase. So, this will naturally bring about a lot of changes in the plant system, in the cells, as well as the genetic makeup of the plants. So, this is, so we have to develop good technologies to study all these aspects, how this affects the plant system. So, first we'll see about the microgravity. As we know, we have heard the microgravity can affect the secondary metabolite production system in the plants. It can bring about growth morphogenesis. It can be elongation stimulated, or maybe lateral expansion may be suppressed, and it will bring about changes in the cell wall. It will bring about the major change will be in the physiological transport. We know that the things will be transported in the plant system through the pull of gravity also. So, this will affect the plant system a lot. So, there are a lot of areas where we are lacking information with respect to the effect of microgravity and radiation on the plant system. First is the long-term effect on the fundamental plant processes, then physiological transport and exchange, then impacts on plant metabolism, then effects on root zone and rhizosphere. So, coming to the effect of radiation, the radiation as such have got, we can take it as in two ways, that we have got both positive effects as well as negative effects. Positive effects in the sense that it has been proven that many of the ionizing radiations are capable of producing genetic changes in the plant system. So, if we are able to fix these positive changes in the plants, we can produce new variants and we can release space-bred crop varieties. And coming to the negative effects, naturally, this will be reducing the germination, it will reduce the vigor, it will produce that, it will affect the growth and produce growth abnormalities and produce many lethal mutations as well. That is causing the sublethal growth changes and subvital mutations can also happen. So, coming to mutations, we know that mutation is a sudden heritable change, and so this mutation can happen both ways. It can be spontaneous mutation or induced mutation. We can harness the positive effect of these mutations through mutation breeding. That is, we'll be developing new varieties by screening out the positive mutants by screening the mutant population. So, this, it is the classification of mutagens, physical mutagens and chemical mutagens. And among the physical mutagens, all these ionizing radiations as well as the non-ionizing radiations are capable of producing mutational changes in the plant. So, as we know, we are not actually as sure what kind of radiations are available in the space. But anyhow, we'll feel that these radiations will can also produce a positive genetic change in the plant system. So the ionizing radiations affect the plant system in different ways. It produces biochemical changes. It produces energy transfer, ionization, and excitation. And low energy transfer radiation can also bring about some destruction in the presence of oxygen, etc. Many of the changes that is happening in the plant system, the plants have a de novo effect of it to tackle that changes. Okay, so through cell repair mechanism, DNA repair mechanism, the plants are, or the plant system is able to go out of this obstructions caused by the radiations. Okay, now with the effect of these radiations, the new branch of science have been developed, that is space breeding. That is, we are using the cosmic radiations present in the space to breed a new variety. And these rare mutants we are obtaining, we can release it as a space-bred variety. And this is what just a schematic representation. For that, when we send the seed lots to the space, we will be keeping a ground control also, and then after germination and then growing the plants for 2 generations, that is SP1 and SP2, we might be able to locate the favorable mutations. So in China, we know this has been started in 1987, and they had been doing this space breeding for more than 35 years, and they have bred a large number of varieties. They have even released a space rice, and they have named it as a rice from heaven, since it was obtained from space. And it has been reported that these have revolutionized the yield outcome from the rice varieties developed by China. So with this background, we have proposed this project. We have 3 aspects we are dealing. First one is exposure of seeds to space. For this, after exposure to the space, we will be studying the, as I have mentioned, we'll be studying the SP1 and SP2 generations. Usually the seed mutations usually occurs at the recessive loci, so it will be expressed only in the SP2 generations. So only by screening the SP2 generations, we'll be able to locate the favorable mutants, and these will be used for confirming the genetic changes through molecular techniques, and we can develop varieties. Then next is the plant growth morphogenesis under change gravity. So we'll be exposing the calli developed of different medicinal plants as well as many flowering plants. So our idea is that after exposure, this callus might show some changes in the cellular metabolic changes, and so this may lead to the may trigger certain cycles and may lead to the production of new secondary metabolites. So we are aiming at that. Then last one, we try to expose the pollen grains. So pollens, as you know, they are haploid in nature, and so they have a better chance of getting mutated. So we will use these pollens after exposure to space for hybridization, and then those seeds will be carried forward, and we'll look for the changes happening after mutation. Then, as we are saying this, we'll be mostly concentrating on the self-pollinated plants. Hopefully you know that, because only then we'll be able to fix those genetic changes. Then this is the proposed plan. Then the seed material will be used for different germination tests. Then seed vigor index tests. Then we'll also be doing physiological steps. For seed mutagenesis, we are planning to send the seeds of the rice, pulses, vegetable seeds, mostly self-pollinated crops. Then the major challenge for us is that in order to maintain these seeds in the viable condition, we need a low temperature maintenance, and also the humidity should be maintained. Then these are the physiological parameters we propose to. Then this is the cytological studies we propose. We are also expecting some ploidy changes or some chromosomal aberrations that may lead to this space environment. Then This is the somatic embryos. So there are reports that these certain callus, when exposed to, that somatic embryos are the artificial seeds developed from the calli. So when the callus is exposed to space environment, there is a higher frequency of somatic embryo production. So we are also checking whether such things are happening. And then medicinal plants usually have the secondary metabolites which are of high commercial value. So when we expose them to the space environment, there is a chance that they might produce some other secondary metabolites which are of more value. Okay, this is the third experiment. Okay, these are the third experiment I have mentioned, that we'll be carrying vials of the pollen grains. Then we did some preliminary experiments at the Kerala Agriculture University. We had a collaboration with VSSC Trivandrum, and we had we planned to take some seed materials, callus cultures, etc. And as a preliminary experiment, we had done some experiment on the calli and seeds at different irradiation levels. But we are not actually sure what level will be available in the space environment. And then we also tried to develop a biopan with the help, you can see this last one. So here we were keeping the seeds in these cabins, and here we took the callus in these tubes, and they were sealed, and we had done the thermal shock test, thermo vacuum test, vibration test, shock test, etc. And these tests were done by the VSSC people, and after that, the scientists of the VSSC, they had handed over the material after these tests, and we had validated them. And we found that the germination percentage was reduced, but we cannot actually conclusively say this, because there was a time span between the experiment and our experiment. Okay. So this project was actually initiated in 2009, and we thought of doing it along with SRE2, but it was called off and we couldn't. So we are still eager of what space has to offer for agriculture science community in the future. Okay, thank you. So once more, these are our team of investigators, and we have the physiologists, we have agronomists, and we have a co-investigator from the NITK Suratkal. He's from the Department of Mechanical Engineering, and he will be the scientist who will be helping us to develop the payload and the biopan structuring, etc. That is the biggest challenge for us. Thank you once again.
Okay, we have time for a few questions. Yes.
Ma'am, won't the mutations caused due to exposing plants to ionizing radiations be random, or will we be able to select the mutations that happen, ma'am?
No, that is actually the mutation that, as I have mentioned, if we expose some 1,000 plants and then we generate, each plant will be giving a large number of seeds, and these mutations will be very minute and will be affecting only a single loss site. And so it will be expressed only in the next generation. So from 1,000, we can multiply the seeds, and we'll have to screen about 10,000 plants to locate 1 or 2 favorable mutants.
Yeah, I think there's one in the middle.
Ma'am, so how frequent are spontaneous mutations during this course? Because then it would somehow affect our results if we are trying to induce mutations via...
No, spontaneous mutations very, very, very rarely occurs. Okay, so there is no chance of getting any mutation in between.
Thank you.
Okay.
Yeah, right here.
Ma'am, the radiation-based mutations could be tested on the ground also.
Yeah.
Instead of, so is there any study, on your side has been done?
Yes, through mutation breeding, we have developed a lot of varieties. At the national level, as well as from our institution, we have developed using gamma radiations and using even chemical mutagens. We have developed a number of varieties. So mutation breeding is a usual process that is done by all the, agriculture
So, advantage of going to the space, I mean, is it because the G-induced mutations might not be, I mean, is it that the microgravity part is also inducing the mutations?
Yes. So actually we are not aware, so we are hoping that since we have reports from China, we hopefully say that that we might get some good mutants.
Okay, thank you.
Okay, so let's thank Professor Jayalekshmy again. So with that, we come to the end of the session.